During my UG, during my PG times, as sir is saying, understanding MHC in a trivial level or trivial manner was fine, but I was not able to understand the whole of it. So I have taken enough time to prepare this PowerPoint. So when we are going through each slide, if you are concentrating it well, I'm 100% sure that you will understand the molecular essence of the whole antigen presentation and antigen elimination with the help of MHC. So before I begin, is there any queries, any doubts you have? You could always type in the chat box. A disclaimer, don't blame me if there is any confusion caused during this session because it will be purely intentional. I would definitely go into the core aspects of the molecular mechanisms. And of course, no PGs were harmed in making this PowerPoint. And in Vivas, I'm sure you all would be questioned. Definitely, there will be a question about MHC. And in university examinations, in theory, it is going to be a essay, antigen presentation on MHC. Sometimes it would be asked in short notes. For sure, there could be innumerable reasoning out questions that can be asked from this topic. So I wish you follow each and everything what I'm just going to say. And make sure you don't quote me in the exams for whatever jokes I'm just trying to. And whatever that is there is enough. It is 99% scientifically accurate. So you just make sure you listen to. And finally, I don't have the intellectual property rights for the language I'm using, the English alphabets I'm using, and even the content of microbiology I'm just going to speak, especially immunology topics. It is all from the standard reference textbooks like QB, Stites, and Abbas. So with this disclaimer, let us move on to today's session. Objectives of today's session, we will discuss about how antigens are recognized by the T lymphocytes, and we will go into the stepwise approach, what happens to the microbial protein antigens, how it is captured by what, here it is in case antigen presenting cell, and finally what happens to those proteins, what is the final fate, what does the antigen presenting cell do, what is the role of B cells and T cells in it? Where are these antigen presenting cells present? And how this antigen would be eliminated from the host. So all these aspects we will discuss about the capture, the processing and elimination of antigen, foreign antigen. And of course, we would discuss about the structure of major histocompatibility complex. We will also discuss about the function of major histocompatibility complex. Can anyone tell me how to remove this yellow line? Sir, are you online? Sundra sir? Maybe I have to use a eraser? Yeah, got it. Fine. So we will discuss about the structure and function of the major histocompatibility complex. We will discuss about the processing and presentation of antigens. And finally, we will discuss about the role of antigen presenting cells. Apart from the display of antigens to the lymphocytes, what does these APCs do? Last but not the least, we will also discuss about the antigen recognition by B cells and other lymphocytes. So let us go in depth about each. So please listen carefully. Antigen recognition by T lymphocytes. Adaptive immune responses are initiated by recognition of antigen receptors of lymphocytes. Adaptive immune response. I believe you all know that immune response is a protective mechanism. Sometimes it could also be harmful, say in case of autoimmune diseases, in case of hypersensitivity reaction. Otherwise, whenever we encounter a foreign antigen, say a microbial pathogen, which is a protein antigen, our adaptive immunity also plays a very important role in final elimination of the pathogen. And we all know that the native immunity that includes toll-like receptors, pattern recognition receptors, the complement system, the cytokine production, all those things, they are part of the native immunity. even your barriers, physical barriers, mucosal barriers, say skin, say mucosal membranes, everything plays part in the native immunity, natural immunity. But when it comes to adaptive immune response, we know that whenever we encounter a foreign antigen, there are certain specialized immune surveillance system that is in place in the name of antigen presenting cells, in the name of T lymphocytes, in the name of B lymphocytes, in the name of natural killer cells, in the name of gamma T cells, etc. So now we will see what happens once we recognize this antigen. The B and T lymphocytes, they differ in the type of antigens they recognize. If you remember in your undergraduate year, you all will remember about MHC and MHC restriction also. Especially when we talk about T lymphocytes, we would discuss about two subsets. One is CD4 and one more is CD8 T cells. And the MHC that it is going to present to is going to be specific for each of the CD4 and CD8 plus T cells. So we'll discuss about that in detail later slides. So the antigen receptors of B lymphocytes, namely membrane-bound antibodies, this can recognize variety of macromolecules. You have to understand it is very, very important. Please remember in adaptive or acquired immunity or immune response, the T lymphocytes generally they take care of the peptide antigens, the protein in nature, peptide antigens. Whereas B lymphocytes, they don't care whether it is protein or carbohydrate or say amino acid or nucleic acid or say lipids or say some chemicals, metals. This B lymphocytes are universal killers. It is like Sehwag going through any bowler, internationally great bowler in the test series. Right? He will be going on hitting fours and sixes. Am I right? B cells are like that, more aggressive. Unlike T cells, which will definitely differ only the peptide and protein nature of the antigens. Whereas B lymphocytes would counter all the protein, carbohydrates, lipids, and even the nucleic acid, even the other chemicals and metal ions. So please remember this. This is very important. This is the basic essence of acquired immunity that you should understand. So these proteins, lipids, polysaccharides, nucleic acids, either in soluble form or insoluble form, as well as the small chemicals and metal ions. We should understand that T cell leads to cell-mediated immunity, whereas B cells leads to humoral immunity. Am I right? Now, before we go into further discussion about this particular slide, let me narrate a story for you. I hope you're all listening. Yes, if you're listening, can you type an S or yes, something like that in the chat box? May I see who is there? Hello. So let me begin with a story. The story goes like this. A thief enters into a busy market. The market is full of peasants, mobs, the people. This thief was planning for a huge burglary. So what he did, he just moved into the crowd and he was trying to steal something more valuable. And one person in the mob, it happens like in Tamil and Kannada movies or any other movies, we see that when a thief tries to steal, a hero comes into picture. Someone in the public will become the hero of the day. Am I right? What they do, they will try to get catch hold of the thief, if possible, give them some beatings. And what would they next do? For sure, they would submit this thief along with the act, the information about the act, and they will submit the thief to the police. Am I right? And once police knows that this is the one who has done this particular mischief, police is not going to kiss or hug him. Am I right? Definitely, police is going to give some dishum. After which, are the police going to give the final judgment? Would they execute the thief? What do you think? Yes or no? The mob in the market caught hold of the thief. The thief was caught red-handed and handed over to the police. Now police is given some beating. Do you expect the police to finally eliminate or kill the thief? Can he pass a death sentence? He cannot, right? He is supposed to submit the thief to the court, right? Finally, the court gives the final verdict or the judgment for the act. So if you take this story and compare this to the immune system and the immunology or the acquired immunity to be specific, you will understand antigen presentation very easily with this story. The thief is nothing but the foreign antigen, the microbial antigen that the host encounters. The mob or group of people who were there in the market, right? They were the one who found out the thief, right? And they caught him, just handed him over to the police, right? The mob is the antigen presenting cells. I believe you all know that antigen presenting cells are present in various locations of our body. You know that in the liver we have Kupffer cells, in the kidney we have mesangial cells, in the bone you have the osteoclasts, in the skin you have the Langerhans cells, the dendritic cells, in the blood you have the monocytes. If you see the tissues, you have the macrophages. Sometimes you have the wandering macrophages that are called as histiocytes. In the placenta you have half-bar cells. In the brain you have the microglial cells, astrocytes, oligodendrocytes, etc. So what I'm trying to say is antigen presenting cells serve different function and they are placed in different locations of our body. It all depends on how a particular foreign antigen, a microbe that is trying to establish infection in the host, is going to enter the body. So it all depends on the mode of transmission. Suppose the microbe wants to enter via skin. There is a damage or a breach in the physical barrier of the skin, say the epithelia. Now the pathogen has access because the common cells of the skin are removed because of the breach on the integrity in the epithelia. So this pathogen would now move into the host happily and would further replicate inside, would invade further, cause symptoms and signs of the infectious disease and infection. Am I right? That's how it works. You should understand that if this pathogen gains entry via skin, say the epithelium or the subepithelia, you should understand that the Langerhans cells or the dendritic cells are the ones that is going to take care of this foreign antigen. So they are the principal antigen presenting cells. They are the predominant, most efficient and professional antigen presenting cells compared to all the APCs. So I now believe that you understand that there are many antigen presenting cells specific to sites which their main function is to catch hold of the foreign antigen. Okay, so we'll again continue the story. I told you the thief is nothing but the foreign antigen, the microbial antigen. I told you the mob is nothing but the antigen presenting cells, and the police, that is our T lymphocytes. You know what this antigen presenting cells do? Once they catch hold of the antigens, they will try to present to the T cells. Sometimes avoiding T cells, they can directly present it to the B lymphocytes. So you should understand that even B lymphocytes can directly counter the antigens without even involving the antigen presenting cells and T lymphocytes. Now I have told you a different model presentation of how antigens could be presented. One, antigens are captured by the antigen presenting cells with the help of a specific particular class of major histocompatibility complex. It will try to present this antigen to the T lymphocytes. Now the T lymphocyte is going to produce some chemicals, say cytokines, interleukins, prostacyclins, say tumor necrosis factor, interferon gamma, all those things which would further damage the antigen. But it is still not going to completely kill it and eliminate it. So what it does is it is going to send this damaged antigens further to the B lymphocytes for the final judgment. So in the whole story, the thief is the foreign antigen, the mob is the antigen presenting cells, police is our T lymphocyte, and the judge is the B lymphocyte. So with this simple story, we have understood antigen presentation. This is the classical pattern. Now I have told you two more miscellaneous ways of antigen presentation, where I told you the antigen presenting cells can capture the antigen and bypass the T lymphocyte. Directly it can present to the B lymphocytes. We know what will happen to the B lymphocytes. The B lymphocytes, once it is naive or immature, on contact with the foreign antigen, it will get activated. It will proliferate. It will mature into a plasma cell. The plasma cell in turn will produce all the immunoglobulins, antibodies. You know that there are 5 different types of immunoglobulins. Am I right? IgG, IgM, IgA, IgD, IgE. Right? Depending on whether it is acute infection, depending on whether the pathogen requires CMI or humoral immunity. Accordingly, the T cells and the B cells would play their part. You understand? It is not the same for everything. Let me also tell you, we are speaking of microbes in general. Whenever it is a bacteria, mostly it is the CMI or the humoral immunity that comes into play. So either T cells or B cells will be involved. Sometimes, not sometimes, I mean, most of the cases, both T cells and B cells have the role to play in the antigen elimination. But there are cases where the predominance of cell-mediated immunity will be more compared to the humoral immunity, especially in case of intracellular pathogens. Say, for example, you take TB, you take leprosy, you take HIV. What happens is all this bacteria, virus, these pathogens, they enter into the cell. Am I right? So they are intracellular. When something is intracellular, the antibodies produced by B cells are not going to work. The antibodies produced by B cells are not going to work. So it has to be the cell-mediated immunity that is going to target this antigens which has infiltrated inside the cell. So please understand that whenever there is an intracellular pathogen, whenever you read about intracellular pathogens, the most common lymphocyte that is going to come into role is going to be a T lymphocyte. But assume that in plasma, in blood, in body fluids, it is mostly the B lymphocytes. Okay, say blood and tissues, it is the B lymphocytes that directly target the antigen. Okay, so with this little notion and story, let us continue further. So the antigen receptors of most T lymphocytes, on the other hand, can see only peptide fragments of microbial proteins. I've told you about this earlier. I'll try to explain it again to you. Please understand that only if the microbial antigen is protein in nature, that is, it is made up of peptides, or when the protein is broken down, it is going to degrade into peptides. Only then the T lymphocytes could sense, see the peptides and could receive these peptides from the antigen presenting cell with help of the specific major histocompatibility complex. But in case of B lymphocytes, I've told you the nature of microbial antigen could be either protein or polysaccharides or lipids or chemicals or even metal ions. Hope I have clarified. Now let us move to the next. So only when these peptides are presented by the specialized molecules that bind peptides generated inside host cell, only then they can display on the cell surface. Here we are in general speaking about the role of antigen presenting cells and the strict need of major histocompatibility complex, which are the specialized molecules that is going to present in the cell membrane, and that is the one that is going to hand over the processed antigen to the T lymphocytes. Okay, so therefore T cell mediated immune responses may be generated only against protein antigen that are either produced inside the cell or even taken up by the host cell. Now, the induction of immune response by antigens is highly orchestrated. I told you it's a specialized immunological surveillance system that our immune system has, which is highly orchestrated process. You please understand this second point. The first is that very few naive lymphocytes are specific for any one antigen. Please remember that there could be infinity of antigens that could attack us. Am I right? We know thousands of bacteria, virus, fungi, species, genus, etc. Assume how our immune system is so complex and is able to handle any type of pathogen. Please understand that the individuals, the composition of MHC is unique. It is not the same. It is always 25% chance, even among the identical twins, to have a same set of MHC genes. So what I'm saying here is only a very few naive lymphocyte. We call a naive lymphocyte as a virgin lymphocyte. It has never encountered a foreign antigen. In that case, it is called naive lymphocyte. So these lymphocytes, when it encounters the foreign antigen, say a microbial pathogen, and only if it is specific for the pathogen, it can encounter the antigen. So you have to understand that among the total 1 lakh or 10 lakh circulating lymphocytes, only one out of them would be more apt. It is like a lock and key. Whatever foreign antigen that is encountered by the body, please remember, though we have 10 power 5 or 10 power 6 of circulating T lymphocytes, only one out of them would have a specific repertoire for the antigen to bind. Please understand, they could only present the antigen when a very rare circumstances, but it is not all or none phenomenon. It is definitely all the time. At least one of the MHC is equipped to handle the foreign antigen. That is how whatever new infection we are encountering, still our immune system is able to respond to it, and there is a difference between the individuals in the way we respond to the infections. Certain people who have those unique specific MHC against the antigen would handle the infection well, would come out of the disease very soon, compared to certain individuals whose MHC would be little different and unique from the other individual, which I told before. So they would succumb to the infection, or they could have the infection for a longer period of time. That could also result in the susceptibility to the disease, sometimes even for the drug resistance. Now, there is a small fraction for the body's lymphocytes that needs to relocate and react rapidly to the antigen, because this is just a small fraction. Am I right? Please understand that wherever they are present, we told that MHC could be present either in the tissues, MHC could be present even in the solution, cytosols, so endoplasmic reticulum, in all those places. Please understand that the small fraction of lymphocytes would definitely come into act against the antigen. Now, secondly, different types of adaptive immune responses are required to defend against different types of microbes. This is what I was trying to explain to you for a long time. There is uniqueness in the MHC subsets, in the genes, in the molecules we have, that perfectly fits as a lock and key against the antigen whenever required. The time taken for the T lymphocytes to react once the MHC starts presenting would be from hours to few days. Minimum of 18 hours it takes for the T lymphocytes to recognize that there is a MHC coded antigen presenting cell that is displaying the captured antigen. It takes nearly 18 hours minimum to a few days for T lymphocyte to act and counter the foreign antigen. So please remember that. I'm sorry, my throat has cracked. I'm sorry for the harshness in voice. Now, assume that there is a virus that has entered the circulation and is free in the blood. It all depends on whether there are antibodies in the blood. I've told you that B cells will always come into play whenever it is the infection or the antigen is in the blood. Am I right? So when B cells come into play, we know that it's going to produce specific antibodies. So the antibodies specific to the antigen, which are present in the blood, they will bind the microbe and it will prevent it from further. It will also try to kill them and eliminate them. Am I right? So this is how a defense against microbe occurs. Say for example, virus has entered blood, the antibody specific to it will react and eliminate them. That is what we are trying to say. Oh, however, the microbe is safe from antibodies which cannot enter the cells. We have discussed this part already. I have told you that antibodies that are produced by B cells, that is mature B cells, that is plasma cells, these antibodies can act efficiently and definitely kill the antigens only if it is present in the fluid compartment, say blood. But if the microbes are hidden inside the cells, these antibodies are never gonna get those microbes. and they can never eliminate the foreign antigen. That is where the CD8 plus lymphocytes, that is cytotoxic T lymphocytes, come into play. You should understand that the T cells, there are generally two types. Am I right? CD4 plus and CD8 plus. So CD4 plus are nothing but the T helper cells, whereas CD8 plus is the cytotoxic or suppressor T lymphocytes. We should know that the CTL, which is nothing but cytotoxic T lymphocytes, they are our saviors, especially in case of intracellular pathogens. If the microbial pathogens are infecting the tissues, they are inside the cell, it is our CTL, that is cytotoxic T lymphocytes, which is a CD8 plus lymphocyte, that is the savior for us, which is going to eliminate the foreign antigen. And now, let me ask you all one question. How do this rare lymphocytes, I told you 1 in 10 power 5 or 1 in 10 power 6, that is going to be the savior. Am I right? So, how do the rare lymphocytes specific for microbial antigen find that microbe? Because it is just 1 in 10 power 5. We don't know where it is present. And where could the microbe could have entered the body? Just think of what you feel. Again, one more question to you. How do different types of immune cells and molecules recognize microbe in different locations such that the helper T cells and antibodies respond to extracellular microbes and CTL kill infected cells harboring microbes in cytoplasm? We have just now discussed the answer for the both. Okay, let me sum up the answer. I've already told you that our immune system is highly specialized for capturing any type of antigen. If it is going to be protein antigen, it is mainly going to be the T cell work, T lymphocytes work. If it is carbohydrate, protein, lipid, or nucleic acid, or chemicals, or metal ions, it could be the role of the B lymphocytes. So what happens once the antigen is captured? What is the next step is that there are going to be some proteolytic enzymes that is going to act on the antigens, would bombard them, beat them, and would break them down into peptides. Finally, these peptides are the ones which would go and fix into the MHC. There is a peptide binding cleft. When I'm covering about MHC, I'll try to discuss about that. That is where the peptides goes and binds to peptide binding cleft of the MHC molecule, whether it is class 1 or class 2. We'll discuss about the structure later. Now, this MHC is the one that is going to display the antigen for recognition to the T lymphocytes, sometimes to the B lymphocytes. But whatever we are discussing, it is so interesting, don't you think so? I wish to thank the research led by the immunologist, the cell biologist, and the biochemist and the microbiologist who are behind all this novel work. It is so interesting and fascinating to understand. Generally, people say immunology is dry. It is never the case. Once you start having passion, try to read in depth. It goes on. Now, let us discuss about the antigen recognition by T lymphocytes a little bit. The majority of T lymphocytes recognize the peptide antigens that are bound to and displayed by the major histocompatibility complex molecules of the APC. We have discussed. And now let us little bit discuss about MHC. What is this MHC, major histocompatibility complex? In simple terms, it is the genetic locus whose principal protein products, they are going to function as peptide display molecules. So MHC, the main role of MHC is displaying the antigen to the T lymphocytes for future elimination. So the MHC has protein components that helps in the peptide display. Now, please understand, in every individual, the different clones of CD4 plus and CD8 plus T cells can see peptides only when these peptides are displayed by the individual MHC molecules. We would have heard about MHC restriction. Let me repeat in simple terms what MHC restriction is. The antigen presenting cell with class 1 MHC molecule can present the capture antigen and display them to the T lymphocytes, but the T lymphocytes are specific. It has to be T CD8 lymphocytes. I'll repeat again. If it is CD8 plus T cells, only class 1 MHC can present the antigen. If it is CD4 helper T cell, it is the class 2 MHC molecule that presents and displays the antigen so that the CD4 molecule will now, CD4 lymphocyte would now recognize the peptides that is displayed by the MHC, which are present in the membrane of the antigen presenting cell. So if we see the overall picture, there is a foreign antigen that is captured by the antigen presenting cell, and the inside there is MHC. This MHC would now move to the membrane and would take the peptides and will now present to the T lymphocytes. And we should know that there is a specific receptor in MHC which will present the peptides to the specific receptor that is present in the T lymphocyte. The lymphocyte of T lymphocytes, the receptor of T lymphocyte, we know it is called as T cell receptor, TCR. TCR. It is different from TLR. Toll-like receptors, pathogen recognition receptors, they are all part of native immunity. Don't confuse TCR with TLRs. Now, let us see this picture. If you see this picture, this rectangle one, this is the peptide or the microbial protein component, and this is the antigen presenting cell. This is the T lymphocyte. If you see the configuration, it is complex, where the T cell receptor, it recognizes a particular class of MHC. I've told you, if it is CD4 T cell, it can recognize class 2 MHC. If it is CD8 suppressor or cytotoxic T cell, it can recognize, the MHC class 1 can recognize and present to it. So please remember. So I'll repeat again. Class 2 MHC can present the antigens to the CD4 cell. Class 1 MHC can present the antigen to the CD8 T cell. Now what I want you to see is that there are some anchor residues. Say this is the peptide. If you see, there are some anchor residues of peptide which are enclosed by using a pocket. So this MHC that is present on the membrane of the antigen presenting cell, they become like a pocket that encloses the anchor residue of the peptide. Okay, so now it is trying to hold the peptide, and now once the T cell is recognizing this particular MHC, now the T cell can collect the peptide via T cell receptor. You understood? So this is how the antigens are recognized, how it is presented, how it is transferred further from the antigen presenting cell to the T lymphocytes for the further process of elimination. Next. The cells that capture microbial antigens and display them for recognition by T lymphocytes, they are the antigen presenting cells. I believe you all would know now. I've already told you what are all the antigen presenting cells that are present at different sites. And I've already told you that dendritic cells among all other antigen presenting cells can be declared as professional antigen presenting cells because they are highly efficient and they are the specialized forms. These dendritic cells are present in the skin, majority, in the epithelia, in the subepithelia. These dendritic cells, say for example, there is a microbe that infects the skin. So it is trying to gain entry via the skin. The dendritic cells which are present in the epithelia, it will catch hold and capture the antigen. And now this dendritic cell which are actually present in the site, they are very immature. They are very early stage immature dendritic cells whose main function is capture the antigen, send it for display with help of MHC. What happens is all the skin, the epithelium, say this tissue, there is going to be a draining, lymphatic drainage. Am I right? It is going to drain to the nearby lymphatic drainage, finally it will go to the lymph node. Am I right? That is how all the pathogens are treated. So what this antigen presenting cell, dendritic cell do is, this immature dendritic cell will catch hold of the antigen, and now it will send this antigen further to the lymphatic drainage via the lymphatic vessel. And finally, it will go and reach the lymph node, which is the peripheral lymphoid organ. Lymph node, spleen, they are all the peripheral lymphoid organ. Whereas bone marrow, thymus, they are all the central lymphoid organs. I believe you all remember the basics. Okay, so let me proceed further. Once this antigen from the immature dendritic cell reaches the lymphatic, lymph node, again there are specialized cells, dendritic cells. These dendritic cells now will capture this antigen, and now this will help the T lymphocyte to recognize for further elimination. So the difference is the immature dendritic cells, they are for antigen presentation, whereas the mature dendritic cells that are present in the lymph node, their main role, it is to allow the T lymphocytes to recognize the antigen. Hope you have understood. Okay, next. These dendritic cells are of two types. One, we call them as classical dendritic cells. Classical. The other one is known as plasmacytoid dendritic cells. Those that are present in the skin, epithelia, etc., they are in the tissues basically. They are called as classical dendritic cells. There is a next type called plasmacytoid dendritic cells. Because of the morphological similarity to the plasma, they are given the name as plasmacytoid dendritic cells. They are present in blood mainly, and of course in some tissues. So please remember the of dendritic cells. The classical dendritic cells are present in the tissue, say connective tissue, whereas the plasmacytoid dendritic cells are present in the blood and also in the tissues. In tissues, I told you the immature dendritic cells will catch hold of the antigen via lymphatic vessel. It will finally send to the lymph node, the peripheral lymphoid organ. So the lymph node collects the antigen from the tissue. If you see here, initially the dendritic cell did not have the major histocompatibility complex, but once it reaches the lymph node, now it has the major histocompatibility complex, MHC molecule. Now with this specific MHC molecule, it could further present to the according lymphocyte, either T or B, depending on whether it is class 1 or class 2 MHC. Now let me also discuss. We discussed already about a virus entering blood. Am I right? So for example, a virus antigen enters the bloodstream via the venule in the bloodstream. It enters the circulation and finally reaches the spleen. So these blood-borne antigens, the blood-borne microbial pathogens, are captured by the antigen-presenting cells present in the spleen. You should know that they are called as follicular dendritic cells, FDC, follicular dendritic cells that are present in the spleen. Hope you have understood. Let me move to the next slide. Okay, so this is a picture of the skin. I have told you that the dendritic cell of the skin, also known as Langerhans cells, it is an antigen presenting cell, a professional APC. So this is the dendritic cell, and this is the MHC component that is along with the dendritic cell. It is in the skin. Now, this picture is nothing but they have added antibodies to the dendritic cell, and this has taken up this stain. So this is some histopathology stain where antibodies to the dendritic cells are added. So dendritic cells appears like this, just like a fibrocellular network madi. Yeah. Again, here it is the lymph node. They have added some fluorescent material, fluorescent dye. So what you see in the periphery of these follicles are, it is the B cells. And in the center, you know, it will be full of T cells. And please remember the dendritic cells are also present along with the T cells in the central area. So please remember that in the lymph node, a peripheral lymphoid organ, in the periphery you have the B cells of the follicles. In the center of the follicles, you have the T cell, T lymphocytes, as well as the dendritic cells. Next. So I've discussed about the classical and plasmacytoid dendritic cells. Let us discuss if a question is asked about this. What are the points that you're supposed to be knowing? So the unique surface marker for classical dendritic cell is that it will have high amounts of CD. CD is cluster of differentiation marker. Cluster of differentiation 11C and 11B will be in higher amounts in case of classical dendritic cells. Whereas in case of plasmacytoid dendritic cells, the CD11C is not high but low, but the CD11B is not at all present. It is negative. Whereas it has B220 in higher amounts. So please remember that it might be asked even in your NEET PG questions. What are the surface markers for the classical and the plasmacytoid dendritic cells? Please remember, already I have told you the classical cells are present in the tissue and plasmacytoid is present in the blood and tissue. And if you see the native immunity component, the toll-like receptor expression, toll-like receptors 4, 5 and 8 is high in case of classical dendritic cells. Whereas in case of plasmacytoid, it is entirely different. It is 7 and 9, TLR 7 and 9, that is high in numbers. And if you see the major cytokine that is produced by classical dendritic cell, it is a tumor necrosis factor. It is interleukin 6. It is interleukin 12. Let me make a point now. Till now, we were discussing that in antigen presentation, antigen presenting cells, the major role it is going to present the antigen. We discussed about dendritic cells. Am I right? But here you are seeing that it is also going to produce the cytokines, unique cytokines like tumor necrosis factor, interleukin 6 and interleukin 12, which would in turn activate and make the lymphocytes, T lymphocytes proliferate, B lymphocytes proliferate. So this is one more function of the dendritic cells. In case of plasmacytoid dendritic cells, one neat PG point that you should remember is it produces abundant of type 1 interferons. So if someone asks you what is the source of type 1 interferons, please remember plasmacytoid dendritic cells which are present in the tissues and in the blood. Fine. You should also understand that for capturing the antigen, especially if the antigenic nature is carbohydrate, these dendritic cells, which are APCs, they use a receptor, sorry for the typo, it's called lectin receptor. Please remember that the lectin receptors are the ones used to capture the carbohydrate structure of the microbial pathogen. But it is not the mammalian glycoprotein that it is going to capture. So host mammalian glycoproteins will not be harmed by the dendritic cells. Please remember, it is only the foreign microbial antigens that will be taken serious and would be captured by the dendritic cells. Please remember that these microbes would enter the dendritic cells either by phagocytosis. Suppose so it's like a macrophage, am I right? Macrophage in tissue is the dendritic cell. So what macrophage does is it engulfs the foreign antigen, it phagocytoses them. Am I right? Or there is one more way where the antigens can enter into the dendritic cells. That is by receptor mediated endocytosis through the specific receptor via endocytosis. I'll show the picture in the subsequent slides. The antigen or the microbe would enter the dendritic cell. So there are two mechanisms, one via phagocytosis, the other via receptor mediated endocytosis. Now, we have already discussed about the native immunity component. I will not discuss more again. This point is again very important. Please remember that once the dendritic cell captures the antigen, it gives some signals and that activates the classical dendritic cells. The signal could be in the form of production of a complement, production of a cytokine, production of a interleukin or a tumor necrosis factor, etc. So what happens is once the other co-stimulators or chemical mediators, once they are produced, the classical dendritic cell would now lose its adhesiveness for the epithelia. So please remember, I told you already, I have shown the picture also that from the epithelium, the antigen captured by the immature dendritic cell, now the dendritic cell will move to the lymphatic tissue. Here, we are showing the molecular mechanism what happens. Please remember that once the classical dendritic cell becomes activated via the signals, it will lose its adhesiveness to the epithelia. So from the epithelia, it will try to move via lymphatic vessel to the T cell area of the lymph node. For this to happen, please remember, it will begin to express a chemokine receptor that is called CCR7. Please remember this point. So the dendritic cell would express a chemokine receptor CCR7 after it captures the foreign antigen. Now it loses the adhesiveness to the epithelia, it moves towards the lymph node, especially to the T cell area of the lymph node. That is where I told you there is dendritic cells also in the lymph node. And these chemocytes make the dendritic cell exit the epithelium and finally goes to the lymph nodes. During this process, the immature dendritic cells that are designed to capture antigens are maturing into a capable ones that can stimulate T lymphocytes to recognize the foreign antigen. So please remember the transition. The maturity from immature DC to the mature DC happens via this process. So please remember the CCR7, don't forget. So once the dendritic cell matures, let me tell you, this point is very important. The MHC molecules that is formed generally are unstable. Only if the dendritic cell has caught hold of the foreign antigen, that makes the stable expression of MHC molecules and the co-stimulators. Please remember. So if you see this picture, it is more illustrative. When you see, you will understand. Antigen captured by the dendritic cells. The dendritic cells are activated via lymphatic vessel. It goes to the lymph node to the T cell area. So from immature, it has become a mature dendritic cell. So if you see the net result, microbial protein antigens are encountered with APCs with specific MHCs, which would further present the antigen to the specific T lymphocytes. This is in summary. So let us delve in more into the cells which mainly exhibits the class 2 MHC. This again will be asked by many. There is one simple dictum. Please remember, all nucleated cells, say for example, neutrophils, say leukocytes, say mesenchymal cells, say epithelial cells, all nucleated cells, they express class 1 MHC. Please remember, all nucleated cells express class 1 MHC. Likewise, we have discussed more about dendritic cells already. These dendritic cells, mostly they exhibit class 2 MHC. So class 2 MHC, it is constitutive in case of dendritic cells, even in case of B lymphocytes, even in case of macrophages. Apart from this, the class 2 MHC is predominant even in case of thymic epithelial cells and vascular endothelium. So there are 5 sites where you have class 2 MHC. Let me repeat. Dendritic cells, macrophages, B lymphocytes, thymic epithelial cells, and the vascular endothelium. Now, class 1 MHC, it is present in all nucleated cells. I've told you the examples, right? Epithelium, say mesenchymal cells, epithelium, neutrophil, leukocytes, etc. Okay. Now, I told you that apart from being an antigen presenting cell, capturing and displaying antigen, I told you it is also going to stimulate the stimulators or co-stimulators. If you see here, the native immunity aspect like toll-like receptor ligands, the interferon gamma, especially if you see the T cell, whenever the T cell interacts with the antigen presenting cell, this relationship is like husband and wife. CD40 and CD40 ligand interactions that occurs between the T cell and the antigen presenting cell. It is very unique. When you are asked this question, you are supposed to draw the picture. I would ask you to refer QB for the picture of antigen presentation, MHC, along with the T cell receptor and the specific ligands. Okay. Likewise, for B lymphocytes, there are specific ligands as well. Now, let us come into the structure of MHC proper. It was the wonderful and spectacular discovery of these three scientists, noblemen, Baru Benacerraf, Sean Dowsett, and George D. Snell. So please remember as Snell, Dowsett, Benacerraf. Even UG textbooks, this is how it will be given. These were the people who discovered major histocompatibility complex and their role, especially in the graft rejection, transplant rejection. So please remember that this MHC, it is like a immune key. It is like a gene of the immune system. It is unique. I've already told you, it is specialized, unique, highly capable, and can adapt according to any type of foreign pathogen that the body encounters. So please understand that these were the Nobel laureates who discovered the major histocompatibility complex. Now, this is the picture that shows the structure of major histocompatibility complex, which is otherwise known as human leukocyte antigen, because the antibodies against the antigens were first demonstrated or discovered in the leukocytes of humans. That's why it is otherwise known as human leukocyte antigens. So the other name for MHC is human leukocyte antigen. Now, there is one more picture of MHC, but this is specific to the mouse. It is called HLA in humans and it is called H2 in case of mice. Now, let me tell you, there are class 1 and class 2 MHC molecules. Some authors even consider this part as class 3 MHC molecules. So, certain textbook authors write that there are 3 classes of MHC molecules, but the standard one, you could just go with this. Please remember that it has class 1 specifically and class 2 MHC locus specifically. It is the genes. It is the genes that codes for production of class 2 MHC molecule and class 1 MHC molecule. So this is a genetic locus with specific genes that codes for the production of peptide components that constitutes the class 1 and class 2 MHC. So you should understand what are the various genes. There is DP, DQ, DR, and DM in case of class 2 MHC. And there is a proteasome component, proteasome genes, say TAP1, TAP2. This is also present in the class 2 MHC locus. So the genes that encode for all the production of these proteins and complements and stimulators, it is all from class 2. Coming to class 1 MHC, it is designated as A, B, and C. And now there are two more miscellaneous items. Say, for example, there are genes that encodes the complement proteins. There are gene or genetic locus that encodes the cytokine production. Say, tumor necrosis factor. Okay, all this you should understand. Now, MHC molecules, I have told you that it is already present on the it is a membrane protein present on the antigen presenting cell. The main role is to display the antigens and it will help the T cells to recognize it. So please understand that it is going to be the major determinant that decides on whether a foreign tissue, say for example an organ, a tissue or a graft is accepted or rejected when it is exchanged between individuals. And the similarity of MHC composition is unique mostly in case of inbred twins, in identical twins, but not the otherwise, or not otherwise. So it is the MHC that is present in the tissues that is the major determinant for graft rejection. In simple terms, you know, major histocompatibility complex, histo means tissue. So the MHC are nothing but molecules or the genes that determine the tissue compatibility between individuals. And please understand that graft rejection is never a natural biological phenomenon. It is never natural. We are artificially grafting one tissue from one individual to the other. Okay. Next, the MHC genes and molecules they encode must have evolved to perform other functions also. We have already told that one is graft rejection. The second is the MHC restriction, which I have already told you that class 2 MHC molecules of the antigen presenting cells can present this antigen only to the CD4 cells, not to the CD8, and the vice versa. So this is MHC restriction. So please remember the structure of MHC. Next, we will now in detail discuss about the class 1 and class 2 MHC molecules that were encoded by the class 1 and class 2 MHC genes. If you see on the right side, this is the schematic diagram of class 1 MHC molecule, where you're seeing that there are alpha component and beta component. So there are three hypervariable regions, alpha 1, alpha 2 and alpha 3, and there is one beta 2 microglobulin subunit. And if you see that the amino terminal is between the alpha 1 and beta 2 microglobulin in case of class 1 MHC. In contrast, if you see the class 2 MHC, it is also typically similar to the class 1 MHC. Only thing is there are separate alpha chains and beta chains, and there are two variable regions, alpha 1 and alpha 2 in alpha chain and beta 1 and beta 2 in the beta chain. And the amino terminal is between the alpha 1 and beta 1 here. So when you draw class 1 MHC, make sure you draw alpha 1 first, in the right side alpha 2, and down it is the alpha 3. Finally, it leads to the transmembrane side chain, which is short in nature. That is the extension from the class 1 MHC. Please remember that this alpha 3 is a region where that is the one that is going to bind to the CD8 T cells. Alpha 3 is the region where it is going to bind to the CD8, and between the alpha 1 and alpha 2, this is the peptide binding cleft I was speaking initially. The foreign antigen peptide, it will reach this peptide binding cleft of the MHC, and now when this MHC which are present in the membrane of the APC, when it encounters or comes in contact with T lymphocyte, it shares the peptide with the T cell receptor. So if you see, this is the place where the T cell receptor will come and bind to and catch and collect the antigen. If you see the class 2 MHC, I told you that alpha 3 is the region where the CD8 plus will bind, that is for class 1, whereas here it is for the T4, that is helper T cell, CD4. Also remember that this cleft, it has a groove. It can hold the peptides which has an amino acid residue of 8 to 9. So about 8 to 9 amino acid residues, that peptide alone can, that long peptide alone can be held by the peptide binding cleft of the class 1 MHC. Whereas if we take the class 2 MHC, it is little more. It can hold the amino acid residue length of about 10 to 30 amino acids. So that is a major difference between class 1 and class 2 MHC. So please remember that alpha 1, alpha 2, alpha 3 is present in class 1 MHC, and beta 2 microglobulin would share the amino terminal with the alpha 1 helix. If you come to the class 2 MHC, the amino terminal is between the alpha 1 and beta 1 side chains. And here it is the CD4 helper T cell that this particular class 2 MHC binds to. Now, you should understand one important thing. Whenever we speak about MHC, I've always told about the word specialized, unique. So please remember that it is polymorphic. The MHC genes are polymorphic in nature. They are highly pleomorphic. That is, they have many alleles. There are alleles, right, for genes, for various phenotypic and genotypic representation, right? So there are many different alleles among individuals. If you compare the MHC composition, if you take the HLA alleles for the class 1 MHC molecules, there are about 10,000 HLA alleles for class 1 molecules alone. Just think of the uniqueness, think of the varied or wide difference or variability, hypervariability I would say. And if you take the class 2 MHC molecules, the alleles that are present are more than 3,000. And next, I would little bit stress upon the nomenclature of HLA genes. We have already showed in the picture that class 1 MHC, we are going to designate as HLA A or B or C. Whereas class 2 MHC, we are going to designate them as HLA-DP or DQ or DR. There are further subtypes also, even HLA-DM. So if you see, the polymorphic genes, MHC genes, are very important because different individuals have different MHC composition. One more important thing is codominant expression. What they mean about codominant expression is nothing but you will get the MHC allele from both of your parents. So it is the MHC allele from each parent will be dominant. That's why it is called as codominant expression. Suppose my mother and my father gave birth to me, I would have the dominant allele, MHC allele, from both my parents. Likewise, my son or daughter would have. It goes on. Please understand that the MHC that expresses cell type, that is class 2 MHC, would the APCs would always be the dendritic cells, macrophages, B cells, thymic epithelial cells, or the vascular endothelium. Whereas the class 1 MHC, they would definitely be linked to the nucleated cells like neutrophils, mesenchymal cells, leukocytes, etc. Okay, so this is the peptide and this is the peptide binding groove of the class 2 MHC molecule, and this is the anchor residue. See, this is the peptide. There is an anchor residue that is hanging, which is covered by the major histocompatibility complex groove. Okay, so this is how the peptide binds to the MHC. Now, if you see the broad specificity, many different peptides can bind to. Please listen to this, it's something important. Many different foreign microbial peptides can bind to the same MHC molecule. So this is the MHC molecule, but you see the peptide that binds to which are all different. So to the same MHC molecule, many different peptides can bind. That is one. And each MHC molecule at one time would display only one peptide. Maybe during the course of time, it can definitely allow other peptides also to bind to it. But one time, it is always one peptide. Next, this MHC molecules bind only peptides. I've already told you when we are discussing about MHC, it is always the peptide or the protein component of the antigen that we are dealing with, not the carbohydrates, not the lipids, not the amino acids, nucleic acids, or the chemicals, or the metals. Please remember. These peptides are acquired when the foreign antigen, microbial antigen, has entered into the cell. Because what happens is, once by phagocytosis, say it enters into the endosome or the phagosome or the lysosome, we all know that there are some proteases that are present in the endosomes or the lysosomes. So this is in general called as endocytic vesicle. So in the vesicle contains the proteases, endosome and lysosome proteases, that is going to attack the antigen. Okay, and this microbial protein is now broken down into small peptide of some amino acid residue length. Okay, I have told you that class 1 can hold about 8 to 10 or 8 to 9 amino acid length, whereas class 2 can hold from 10 to 30 amino acid length. That fragment of protein that is processed by endocytosis. Next. So this is what we are discussing. Once the foreign antigen via endocytosis enters into the endosome or lysosome, what happens is this MHC molecule that is present in the endosome, apart from the MHC molecule, it also has one more component that is called II, that is invariant chain. The invariant chain is like keeping a cover for your glass tumbler so that no other dust goes in. So nothing can be say any host peptide can never bind to MHC, and MHC would not act on the host self peptides, which are protein in nature, because it is covered by the invariant chain. This invariant chain has a component called as CLIP, CLIP. Please remember, CLIP. CLIP is nothing but class 2 invariable chain peptide. So when there is a MHC, before it encounters the peptide, it actually has a cap. That is nothing but the invariant chain. This invariant chain consists of something called as CLIP, which is nothing but class 2 invariant chain peptide. Now, when this antigen presenting cell along with the MHC and invariant chain and clip combo, once they go to the lymphatic vessel and go into the lymph node, that is where what happens once the antigen comes into the MHC binding cleft, say peptide binding cleft, this clip or the invariant chain is now comes out and the peptide binds it. After which this MHC along with the presented antigen would now be recognized by the T lymphocyte via the T cell receptor. Whereas there is one more aspect to this of processing the antigen. Instead of by endocytosis, the antigen, say for example a virus is present in the blood in the cytosol. What happens is the cytosol protein is unfolded, and then the cytosol protein will be now combined to something called as ubiquitin. Ubiquitin. Once the ubiquitin bound cytosolic protein, this enters into the proteasome. It gets threaded and shredded. So now the cytosolic protein along with ubiquitin complex, they are broken down into small peptides. Now what happens is there is a unique MHC for cytosolic proteins. I have discussed already. Tissue, it is mostly the MHC class 2. If it is cytosol, mostly the class of MHC is MHC 1. What you should understand is class 2 MHC had something called as invariant chain which had clip. Whereas if you take class 1 MHC, it is connected to something called as TAP, which is transporter associated with antigen processing. I'll repeat again. TAP is nothing but transporter that is associated with antigen processing. So this transporter associated with antigen processing is linked to the class 1 MHC by something called as tapacin. So please remember, when we are discussing about antigen presentation with class 2 MHC, there you should remember about invariant chain II and clip, that is class 2 invariant chain linked peptide. When it comes to class 1 MHC antigen presentation, please remember that it is not inside the tissue, it is inside that cytosol. And there is something called ubiquitin that will form a complex with the cytosolic protein. It gets unfolded. Finally, it is threaded via an apparatus called as proteasome. It is a proteolytic apparatus, proteolytic structure. That is proteasome. The proteasome in turn contains some proteases or lytic enzymes that is going to break down the protein into peptides. And I said in class 1 MHC presentation, I said that these MHC molecules are present inside the endoplasmic reticulum and they are bound to TAP, that is transporter associated with antigen processing. The TAP is connected to the MHC class 1 molecule via something called as tapacin, TAPASIN. Now what happens is this peptide that is broken or shredded is caught hold by the TAP protein that is bound to the tapacin in turn to the MHC1. It will leave the endoplasmic reticulum and via it further goes to the CD8 or cytotoxic T lymphocytes and presents the antigen to the T cell receptor specific to the CD8 cell. This is what that happens. This is the two unique pathway of processing of protein antigens. One is endocytic and one more is cytosolic pathway. I hope you all would have understood now. Okay, so this points I've already told. I am not going to explain again. Please remember that in the structure of class 1, it is alpha 1, alpha 2, alpha 3 and 1 beta 2 microglobulin. Whereas if you take the structure of class 2 MHC molecule, it contains two hypervariable chains, alpha and beta, which has alpha 1, beta, alpha 2 and beta 1, beta 2 parts. And I've told you that the amino acid terminal is the area where there will be peptide binding cleft. Here, the amino acid binding area is formed between the, here the amino terminal is the area where it is formed between alpha 1 and beta 2 microglobulin. Whereas here it is formed between the alpha 1 and the alpha 2 amino terminal. This is where you will have the peptide binding cleft and the antigen would bind to it. So please remember that when the antigen is entering the cell via endocytosis, it goes inside the endosomes or phagosomes or the lysosomes. It is internalized. The antigen, protein antigen is internalized from the extracellular environment into the endosomes and lysosomes and phagosomes. And that is going to contain proteases, say for example cathepsins. This is the one that is going to grind and lyse the protein into peptides. So the site where the peptide loading occurs is in the late endosomes and lysosomes. And again, I'll repeat, the molecules that is involved in transport of peptides is the invariant chain II and the clip, that is class 2 invariant chain peptide in case of class 2 molecules, whereas in case of class 1 it is the TAP and tapacin. TAP is transporter associated antigen processing, associated with antigen processing. So these are all those pictures. I think with this we have understood the presentation of protein antigen and the processing and their elimination. Apart from antigen display and the presentation, I have told you that they have their role in production of signal molecules and co-stimulators that would in turn activate the T lymphocyte, which again, it is part of the native immunity. So the complement production, cytokine production, the TLRs, that is toll-like receptors, the pathogen recognition receptors, all those things are the, when they get activated, they are the ones that provide signal so that the APCs would now activate the T lymphocytes. Now, what about the antigen presentation by B cells? We have already discussed that T cell, the antigen recognition, it is mostly going to be the nature of the antigen that decides. So protein or peptide antigens would be recognized by T cells. And already I have told you that initially B cells can recognize peptides or even the whole proteins. See, T lymphocytes can recognize only the processed protein, only the peptides, individual peptide units, only the peptide, broken peptides. That is what is recognized by the T cells. But if you take B cells, the whole microbial antigen, not the peptides, even the whole microbial antigen, let it be the protein or the carbohydrate or the lipid, that is the composition, it can still attack and eliminate them. So I've already told you that the B cell is rich in B cell rich lymphoid follicle of the lymph node. In this area, you have follicular dendritic cells. Please remember that the follicular dendritic cells, they display coated with antibodies or they coat with complement, especially C3b and C3d. Please remember this point. Follicular dendritic cells, please remember that it is present in the B cell rich lymphoid follicle of the lymph node. There is one more follicular dendritic cell that is discussed in spleen. So peripheral lymphoid organs is the area where you would find follicular dendritic cells. So with this, let me conclude by saying that antigen presentation, antigen display, antigen processing, and antigen elimination is taken care by more highly complex and sophisticated infrastructure of immune system that we have. And we are able to tackle any new, if not now, in the past and also in the future. So with this, let me thank the organizers for helping me present this topic to the postgraduates. Thank you so much. If there are any queries, any explanation needed from my side, I'm happy to explain. You could unmute other participants, sir. There's something in the chat box. Let me see. Satyanarayan, sir. Yes. Yes, sir. Okay. So you've told yes. Sorry. Thank you. So is there anything that you want to discuss? You want to ask me? Have you understood how the antigen presentation happens? What is the basic structure and genetic makeup of MHC? And how MHC class 1 presentation is different from class 2? If that has been covered, if you're 100% confident that there is no doubts, my job is done. Thank you so much. Thank you for the opportunity.
Yes, sir. Good evening, sir.
Yes, sir. Good.
I'm sure this recording will be shared with all the PG students and various. I'm 100% sure this will be the most top one recording that will be gone to again and again for PGs.
Yes.
Because and when this topic was discussed for the PG masterclass, even I was
Yes, sir.
worried whom to get to talk this topic. Only a person like Dr. Someshwaran could handle it.
Oh, thank you, sir.
And
Sir,
wonderful work, sir.
I'm just a junior. So there are many stalwarts who are senior professors from whom I have learned all these things from. Of course, from the
Yes,
textbooks as well.
and,
So I wish
and,
to remember them and salute them for all the hard work they have put in, so that they have made me understand all these things. Thank you.
uh, and this is not easy. It requires a lot of dedication, background preparation, and see, I already they say please take more immunology classes.
Sure, sir.
See,
Sure.
the immunology is one thing, sir. Many people, whenever we ask, even if you ask me suddenly, one thing immunology and parasitology, two things, many people don't wish to talk much.
Yeah. True. True. True.
And, and you have made it as simple as possible and given all your 200% efforts into and we can make out how much you have gone to the depth of the subject to prepare for this class. 100%,
Yes, sir.
sir.
Yes,
It
sir.
is going to. I don't think any queries are there because we need to go to again and again to get to the depth of the
Yes, sir.
subject.
Yes, sir.
So,
Sir, with your
but
permission, let me show this IgA ability slide.
I will
Five
say
five
you want.
minutes. Yeah,
Yes, sir.
five
Please,
minutes.
sir. ICG avidity, that is one.
Yeah, just tell me if you are able to see the full slide.
ICG avidity test, no, sir?
Yes,
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 sir.
because you asked me that you told that many PGs wanted to cover this also. So what I have decided is to give a bird's eye view. I'm not going to go in depth, but this is enough for to write a short note. Or when asked about, you could definitely give a beautiful answer. So let me continue with that also, if it is okay, if the time permits.
Yes, sir. Please, sir. ICG avidity test.
Yes, yes. So let us discuss about what is IgG avidity. IgG is immunoglobulin G
Just
and
10
avidity.
minutes.
5 minutes,
You
five
have
minutes
got
max.
PowerPoint, sir?
Yes, yes.
You have got,
Sir,
that is not
I've
displaying,
opened,
sir.
I've opened it. Okay, let me share again.
You close it. You see,
Let me share again. Let me share again. Is it visible now?
it is starting, sir.
Yes, visible?
Yeah, it is visible, sir.
Fine. So in immunology, it is all about the antigen, foreign antigen, and the antibody that is produced in the immune system. When we talk about humoral immunity, B cell mediated immunity, it is about antibodies countering the antigen. Am I right? So when an antigen binds with the antibody, we know we would have heard more about antigen antibody reactions. I believe the PGs would be taking classes, practical classes to the UGs also about antigen antibody reactions. When an antigen antibody reacts, please remember that whole molecule of antigen and antibody has to react, and the reaction is also reversible. It is not irreversible, it is reversible. Please remember. And it is not only a portion of the antigen and antibody that reacts, but the whole antigen and antibody that reacts. Now, when an antigen and antibody reacts, for it to react effectively, it needs an extra person that is called complement. You would have heard of the term opsonization. Once the complement binds to the antigen, helps the antibody neutralize the antigen well, that is called opsonization. When we discuss about toxins, we discuss about neutralization, opsonization, etc. Why I'm discussing about this binding of antigen and antibody and the role of complement is that the complement is like a catalyst. It's a positive catalyst. It allows the antigen and antibody to bind effectively. Okay. Now, let me tell you, we use two terms to define how much is the bond between the antigen and antibody. The words are affinity and avidity. Affinity is like a boy and a girl in, say, a teenage. You have some attraction towards. It is a strength of attraction between the antigen and antibody. That is affinity. I'll repeat again. The strength of attraction between antigen and antibody is the affinity. Whereas once the antigen and antibody with affinity have bound together, now it requires some amount of energy to break the bond between the antigen and antibody to dissociate them and separate them each other. You understand? Affinity is nothing but the strength of the bond that is formed between the antigen and antibody. So the word IgG avidity test simply means we are going to find out the strength of the bond that is formed between the immunoglobulin G and the specific antigen. If the strength is more, there is a clinical significance. If the strength is less, there is a clinical significance. Let me tell you this thing before that. The immunoglobulin G matures over weeks. So early immunoglobulin G, the avidity will not be more. But over period of time, over weeks to months or years, the strength that is required to break this IgG and antigen combination would be more. You understand? So the avidity increases over time period because the IgG matures only over the time period. Now, when you find low avidity, when you do the IgG avidity test and you find low avidity, it means that it is a recent or a early infection. When you have a high avidity, it says that it is because of vaccination or past infection. Am I clear? If there are any doubts, you could stop me here. Now, principle, please remember one thing. How we demonstrate avidity test? Very simple. Antigen is coated in the well. Now, this is exposed to the serum that contains immunoglobulin G. What they do is they add a denaturant which will denature this bond formed between the antibody IgG and the antigen. So we use urea or guanidine. Please remember this. The denaturant used in IgG avidity test commonly are urea or guanidine. So this either urea or guanidine will now disrupt the bond formed between the IgG and antigen. So if there is low avidity, it means that easily the denaturant has done its job. If there is high avidity, it is because the denaturant was not able to break the bound bond. So this avidity index is further measured in terms of optical density, and the calculation goes like this. The OD of the IgG avidity test when done with denaturant, how much, divided by the optical density without using denaturant, how much, multiplied by factor of 100. So this is the formula that is used to calculate something called as avidity index. For doing IgG avidity test, indirect immunofluorescence can be done. Even ELISAs are available for performing IgG avidity test. Please remember this. I have not put in the slides. Please remember this. Indirect immunofluorescence test or ELISAs are the commonly used methods for performing IgG avidity test. Now, if the strength of the bond, that is the avidity, is found to be less than 30% to 40%, less than 40%, it is for sure recent or early infection. If the avidity is found to be more than 60% based on the avidity index, for sure it is a past infection. But when it is an intermediate, there is a gray zone, better to repeat the test after 2 weeks. Okay? How this is important? Please understand that IgM is not reliable marker. IgM start rising early in the infection. It reaches peak after some days to weeks. After some time, it disappears. Again, during the advanced stages, it reappears. So mere presence of IgM does not always say or differentiate early infection from the past infection. That is why IgG avidity test has gained its importance, because it takes time to rise or appear, then it reaches peak and it is persistent for a longer period. And I've told you that the IgG, it matures with time period, and the avidity is going to be different from the recently found IgG or IgG that is there for a longer time. That is the whole idea of using IgG avidity test, and this is mainly critical for pregnancy related infections. I believe you all know torch infections, am I right? Toxoplasma gondii, cytomegalovirus, rubella, herpes virus, herpes simplex, varicella zoster virus causing chickenpox, say parvo B19 virus. So these are all the key pathogens for which IgG avidity test is required as a part of diagnostic microbiology, as a part of diagnostic stewardship, especially in case of pregnant women, where we are trying to rule out there is a risk for congenital infections or not. So please remember TARCH, Toxoplasma gondii, CMV, Rubella, HSV, VZV, and Parvo B19. So for Toxoplasma gondii, it is the most validated test, IgG avidity test. For CMA avidity, generally the IgM as usual is non-reliable. So the high IgG avidity again in trimester especially, if we know that the IgG avidity is high, definitely it is not the recent infection. So we need to terminate the pregnancy. What if it is low IgG avidity? It is very bad news for the patient. The higher chance of early infection in the pregnant woman. For sure, we have to terminate the pregnancy. That is the importance. So same with the rubella. Now, last one, the limitations, as I have told you, the maturity of IgG over time, it varies by pathogen. Again, it varies by time period. Again, in case of immunocompromise, IgG avidity test results are not valid. You cannot, it's not reliable. Again, interlab kit variation, procedural variation could give varied interpretation and misread results. So with this, let me conclude the session. If there are any doubts, I am happy to answer. Thank you, sir.
One question in the chat box, sir.
I will go through. Yes, I have seen some messages. Thank you for, sir. Please take more immunology class. Why not? Why not? Why not? Happy to take. I think 2nd of November, December, I have a class on native immunity and acquired immunity for MBBS students. I don't know whether I can send the link to you. Very high serum urea levels disturb. Excellent question. Excellent question. Definitely. Definitely. So, what I would suggest you, thanks, Mr. Dr. Raghavendra Kulkarni. I wish you try to find a brochure of IgG avidity test, either by immunofluorescence or by ELISA. You please carefully read the troubleshooting guide page, where all those this extraordinary or unpleasant situations or abnormal circumstances are discussed, and the way to deal with is also discussed. Definitely, when the serum urea levels are high, obviously there is going to be problem with the antigen binding to the antibody. Okay, that would definitely. That's why if you see in case of renal failure, the clearance of antigen from the host, it is delayed. Even if you take COVID pandemic, you all would be hearing or would have heard from your nephrologist talking to you, your ID physician talking to you, saying that why doctor there is no clearance, why there is no clearance, why there is no molecular conversion, why there is no zero conversion that is supposed to happen in a particular period of time, just like in normal individuals. In case of immunosuppressed or immunocompromised individuals, your immunity is hampered, your antigen presentation, recognition, elimination, nothing is going to work in the way that you expect to. It all depends on which immunity is compromised, whether it is T cell or the humoral or both. Accordingly, it is going to happen. Okay, so hope I have answered your question. Thank you. Hello?
Thank you, sir.
Yes. Thank you. Sir, shall we end the session?
Dr. Kulkarni, sir? Adi Kulkarni, sir.
Raghavendra Kulkarni, sir.
Hello.
Hi, sir. Good evening.
Hello, good evening. Am I audible?
Yes, sir, you are. You are. But you are not visible.
It's okay.
Tell me, sir.
My logo is visible. Okay.
Yes, sir.
It was an excellent say presentation, and
Means a
thanks.
lot, sir.
You
Thank you.
cleared a lot of concepts. Thank you very much.
Thank you, sir. Thank you, sir. Thank you. Okay, thank you all. Sir, thank you, Sudra sir, so much for giving me the opportunity.